Good evening. I'd like to thank you very much for coming to our Midsummer Night Science lecture tonight. My name is Megan Rokop, and I'm the director of the Educational Outreach Program here at the Broad Institute. It gives me great pleasure to introduce our speaker tonight, Dr. Bill Hahn. Bill is a medical oncologist and an associate professor of medical oncology at the Dana-Farber Cancer Institute. Bill received his bachelor's degree from Harvard and his MD and PhD degrees from Harvard Medical School. Bill currently serves as a senior associate member of the Broad's Cancer Program. And tonight, Bill will be speaking to us about his work in finding the Achilles heels of cancer cells. Please join me in welcoming our speaker, Bill Hahn.
Well, thank you very much. It's great to see all of you, and I'm glad that many of you braved the schizophrenic weather out there to come. So what I wanted to tell you about today is about how we're trying to understand cancer. And the title refers to a Greek myth about a hero called Achilles who unfortunately had this weak spot, which was his heel, and because his mother dipped him in a river that would protect him. And in some ways, the reason to think about that is cancer, in many ways, is a disease that strikes a lot of fear into people. Probably everyone in this room has had someone or known someone or even themselves been affected by cancer. And I think in some ways it seems like, to many people, an invincible beast. But what we're learning is that actually cancer has its weaknesses. And the problem is that it's been very difficult over time to find those weaknesses and exploit them. But I think that there's a lot of reason to think that we're making progress and that we're in a really unique time period now where we can really try to get at what might be Achilles heels of cancer. Now, some of you in the audience have a lot of scientific background, but this lecture is not directed toward you. It's actually directed for people who have a less scientific background. And so I think to start out and get everyone on the same page, we should think I thought we would start out by some definitions. Now, so the first question is, what is cancer? And everybody has their own view of what cancer is. And so there are ways that you can find out information about cancer. This is the old school way. You'd look it up in the dictionary. And there's some, when you look things up in the dictionary, you find interesting things. It could be that cancer is a zodiac sign. That's not the cancer I'll be talking about tonight. It is actually this one, a malignant tumor of potentially unlimited growth that expands locally by invasion and systematically by metastases. That doesn't sound very good. For those of you who are younger or more web-based, we'll look at the Wikipedia definition of cancer. It's a class of diseases or disorders conferred by uncontrolled division of cells and the ability of these cells to invade other tissues, either by direct growth into adjacent tissue or through invasion or implantation to distant sites by metastasis. So strikingly similar, making you think that someone from the younger generation actually just copied and pasted the definition from the other one into Wikipedia. So in any case, that's where we're starting with. And I think the way to think about cancer, one way to think about cancer is it's an abnormal growth in the body that shouldn't be there and that causes problems because it grows into tissues around it. The other way to think about cancer is it a problem for society? And I think it is. And the way to look at this is to say these are data from a few years ago, but we could say, well, what do people in the United States die of each year? Kind of a morbid thought, but important to think about it. And in fact, cancer is number 2 on this list, that more than 500,000 people in 2004 died of cancer. And that's actually quite an incredible number. And I think it goes to say that it is a very important health problem. And I think another way, a more personal way, is what I referred to earlier, is that probably everyone in this room knows somebody affected by cancer or has been affected by cancer themselves. So it is a problem that is one of very clearly of medical importance. You can look at data to say, well, there are many different types of cancers, and some are more prevalent than others. And you can see over time, this goes from 1972 to 2000, for a lot of the cancers, there really hasn't been a change in how many occur. There's this little funniness around prostate cancer, which has to do with a PSA screening test being introduced. And when it was introduced, a lot of people got screened, and we found a lot of cancers. And so this explains this precipitous jump. It isn't that something happened in 1990 that increased the risk of prostate. What we're doing here is catching all the prostate cancers from the previous 20 years in sort of 3 years' time. Now, the bad news about this is, even though that the number of cancers is not really going up, is that these are the death rates for the different cancers. And although in the last few years there's been a reassuring trend for both men and women of cancers going down, far too many people still die of cancer. And I think, well, in both men and women now, lung cancer causes the most deaths. And in men, it's been going down for a couple of decades. Women, unfortunately, because women started smoking in larger numbers more recently, their numbers are still going up, because it takes about 30 years from the time you start smoking to the time you develop lung cancer. So that's another way of defining cancer. The final way I thought we could define cancer is how someone like me, who's trained as a medical oncologist, a physician who takes care of cancer patients, thinks about cancer. And that is, even today, we think about cancer in 4 ways. One is we think about, where did the cancer start? Where is it a lung cancer? Is it a breast cancer? Is it a colon cancer? And then we think about, well, if we figure out where it started, how far has it gotten from the place it started? At some points, we'll have a surgeon or another physician take a biopsy, and we'll look at the cancer under the microscope, and I'll show you some of that to say, what does it look like? And in very rare cases, we have things called markers, like a PSA test or a few other tests, and there are not very many of them. And really, that's it. That's how, in 2008, a medical oncologist at a tertiary care medical center anywhere in the United States or around the world thinks about cancer. Now, why we think about cancer this way as physicians is this allows us to predict prognosis, to make guesses about, educated guesses about what the course or the natural history of the disease will be if we do or do not try to intervene. It guides how we make decisions and present them to patients about their therapeutic options, what's likely to work, what's likely to be effective, what are the possible outcomes. And these markers and this information allow us to, in a larger sense, to determine whether or not we're making progress in cancer. So I've talked a lot about where cancer starts, and I think that this brings up the idea that cancer, although we think of it as one big disease, is actually probably more than 100 diseases that are defined, at least today, by where the cancers start. And they're given various names, like leukemias for cancers in the blood, sarcomas for cancers that come out of fat or muscle or bone, and then epithelial cancers or carcinomas that are the ones that we often hear about in the news: lung cancer, breast cancer, colon cancer, ovarian cancer, and prostate cancer. So when someone is being diagnosed with cancer, the first question that your physicians are trying to figure out is, where did it start? Once they've figured out where it started, they do a lot of imaging and other tests to figure out where the cancer has spread to. And the reason that we want to know how far it spread is because over the decades, physicians have learned that if you figure out how far the cancer has spread from the initial site, you can predict what will happen to the patient over time. And it's a very simple idea here. If the cancer is very localized and it can be cut out by a surgeon or zapped by a radiation oncologist, the chance that you'll be cured of the cancer is much, much higher than if it's figured out how to spread away from the original site and infiltrate into the adjacent tissues and maybe into other organs. And for example, this is colon cancer. If you have a localized colon cancer found by a colonoscopy, and it's very localized and small, you have a greater than 95% chance of being cured of your colon cancer if it's found at that stage. However, if your colon cancer has spread outside of the colon and into lymph nodes, you can see that the chance that the colon cancer is going to cause a real problem and may kill you is much higher. So this is why this is important to do what physicians call staging. It's to figure out how far has the cancer spread. Now, we also take some of the cancer in tissue in a biopsy and look at it. None of you, or very few of you, are probably pathologists. And so this is a little bit of pattern recognition. This would be considered normal colon, and this is abnormal. And it doesn't take someone who's highly trained in pathology to recognize that there are some nice patterns here that are all mixed up over here. And that's what the hallmark of cancer is at a histological level, is that there's the uncontrolled growth of the cells has disrupted the normal patterns that you see in the organs. And that's generally bad. So I have to admit that although this is what we do in the clinic, and if I were in the clinic today, these are the criteria I would use when I see patients, it's a little bit embarrassing. After many, many decades of really studying the molecular basis of cancer, we're still using archaic ways, in my mind, of thinking about cancer and figuring out how we're going to treat patients. Moreover, if we think about what the challenges are, is that we find cancers often when they've spread beyond the initial site. We can't always predict what the outcome is going to be. Sometimes we think that people are going to do very poorly, and occasionally they do better than that. But oftentimes, people do much worse than we hope they will. And then finally, although we have made a lot of progress, we still can't cure most cancers in adults. And so these are stark facts, and they are, and I think given the scope of the cancer problem in the United States and other countries, it behooves us to try to figure out how can we improve on this, and what can we do to bring cancer into the 21st century and understand it better. Okay, so let's now think about cancer from another definition, and that is, let's think about cancer if you're a molecular and cellular biologist. And so if you're a molecular and cellular biologist, your view of the world, or the biological world, is that there are cells, individual units, that cooperate together to make up a normal tissue. And that tissue could be your skin, your heart, your lungs, any organ in your body. And what happens in cancer is that there are normal processes that tell a tissue that we need more cells. If you're growing or you're injured and you have to replace cells, there's a process here to replace these normal cells. And at the same time, there's signals in the normal tissue that say something's wrong, we've got to get rid of some abnormal cells and kill them off. And most of the time, greater than 99% of the time, this system works great. You get rid of the bad cells, you replace them with rejuvenated cells, and you don't grow if you're an adult, and your organs continue to work well. But what happens in cancer is two things occur. One is that somehow there become abnormal signals that say, we need more cells. And so the signals go out to say, we need to make more and more cells. And so in an abnormal way, the tissue now starts making more cells. And at the same time, these control systems that say, something's wrong, we need to get rid of cells, goes haywire. And so there's no alarm system to say that, hey, we have too many cells and we should stop making them and get rid of the ones that are extra. So you can see how these two things lead to the bottleneck of having way too many cells and having them get out of control. So why is it that this happens? And what we've learned over the last 20 or 30 years is that cancer is a disease of the genes. It's a genetic disease. And what I mean by that is that all of us in each of our bodies and each of the cells has DNA, and this DNA arranged in chromosomes is the blueprint for everything that happens in each of your cells. And what happens with this blueprint is that you accumulate mutations in particular pieces of it as you go through your life. And those mutations can come from a variety of ways. Some people will inherit some mutations that will make them predisposed to diseases like cancer. That's actually quite rare. You could be exposed to radiation in the environment. And generally, this is not low doses of radiation. This is large doses of radiation if you happen to have lived near Chernobyl in the 1980s. There are some viruses that can cause cancer, and chemicals is a generic way of saying other insults over the course of your life can damage DNA. Now, normally, there are signals that say the DNA is damaged in a cell, and we should get rid of that cell, and things are fine. But what happens is, over time, some of those control mechanisms go haywire, and so you accumulate mutations over time. Now, there are two types of mutations that are important to think about. Now, this is a funny word. It's called oncogene. And the reason for this word is that it turned out, and this was a big discovery in the 1980s, that the genes that are mutated in cancers, the genes that lead to cancer, are actually mutated versions of our normal genes. And it's important to go back about 30 years ago. It was thought before that time that what happened in cancer was it was a little bit like having an infection, that something came in and added extra stuff that led to the cancer. And while that happens with a few viruses, like the virus that causes cervical cancer, for the most part, cancer is a disease of mutating our own genes that we carry around that have normal functions. And these oncogenes are genes that are the class of genes that tell cells to make more copies of themselves, to have more cells to replace the cells that are missing. And what happens in cancer is that you get mutations in these genes, and that leads to a signal that says we need more cells. The other class of genes that I alluded to were those that said, hey, there's something wrong. We should get rid of cells that are damaged. These are called tumor suppressor genes. And what happens in cancer is that these genes get mutated, and they're no longer active. And this is a way of inactivating those alarm systems that protect us from cancer. Another way of saying this is that tumor suppressor genes are a lot like the brakes on your car. They're there to prevent you from speeding off and crashing into something. But of course, if your brakes fail, it gets very difficult to stop your car. So what I've outlined is that cancer is a disease of having too many cells, and that those cells are not following the right orders, and that the reason we get to that state is that there are mutations in genes. And so the question is, how do we understand how many mutations there are in any given cancer? And the reason I started out the lecture by saying that although we still can't cure most cancers, that we're in a very precipitous stage of scientific inquiry where I think things are going to change rapidly, is that our ability to look at all the genes in the genome has changed dramatically in the last 10 years. Now, one way of thinking about it is if we think about maps of the world. So in the 1400s, this is one of, I don't know, many different types of maps that people drew to say this is what the world looked like. And there were some salient features. Most of the world was unexplored. You could sail off hoping to find new lands and new riches. Many people thought the world was flat. It wasn't round. And I think it's safe to say that the world was mysterious. People would get in their ships and sail off because they thought they'd find something that they couldn't even imagine. And many times they would. Cancer in 1996 was much like this. We knew that it was a disease of genes, and there were mutations in those genes. But the problem was we didn't know how many genes there were. We didn't know what the genes looked like. And therefore, all we had the ability to do in the clinic was to try toxins or poisons to try to kill cancer cells and leave normal cells alone. And therefore, our treatments were largely empiric, designed to kill cells that were growing faster than other cells. And the genome of the cancer cell, then as it is now, was considered chaotic. Now, by the 1990s, our view of the world had changed. There were no more unexplored land masses. You're not going to find a new island that nobody's ever found before. They've all been mapped. The world's now considered to be round. There are a few flat Earth people out there, but generally people believe that the world is round. And by 1990, there were high-resolution maps of every spot on the Earth. So you could see wherever you wanted to see. And that corresponds to 2003, when the human genome was finally sequenced. And what that allows us to now say is that we know the overall structure. Finally, by 2003, we knew the overall structure of the genome. We had sequenced all the bases in the genome. And we knew what the map of where all these genes lived. And it turned out, and it was a big surprise, that there weren't as many genes as everybody thought. When I was in graduate school in the late 80s and early 90s, it was every graduate student's dream that you would clone a gene, you identify a gene, and that you'd be the person who discovered this gene. And all of us thought in graduate school that there were so many genes, millions of them, that we'd be doing this forever. It turned out that there's only about 20,000 genes. And that was a big surprise when they sequenced everything, that there was actually only 20,000 of these things. And now my graduate students, it's inconceivable to them that they would think that they would clone a gene that nobody had ever discovered before, because they've all been discovered. Now, if we fast forward to where we are just about now, maps have gone a dramatic change. Now we have global positioning satellites that allow us to carry around handheld units or even phones that will tell you exactly where you are. And if you want, they'll even give you personalized directions to get from point A to point B. So the question is, if that's the case with maps, what are we doing in terms of genomes and cancer genomes? And I think the nice thing here is that a lot of efforts of investigators at the Broad Institute, as well as international and national and international efforts, are applying technologies, some of them developed here, to be able to essentially do GPS on cancer genomes, meaning that we can take a variety of techniques, the details of which are unimportant, but be able to map all of the mutations that occur in any cancer gene in any tumor, and be able to say, here's the list of things that are mutated in your cancer. And what's very exciting about this is that these efforts are ongoing. There are probably machines running upstairs doing this work right now, as they are around the world. And that I think in the next decade, we will know all the genes that are mutated in any cancer. More exciting than that, these technologies are moving forward to the point where it's possible to imagine that instead of coming to the clinic and being told you have breast cancer or lung cancer, you'll be told that you have breast cancer or lung cancer, and here's the list of things that are mutated in that cancer, your cancer. And so I think in the next decade, what we're going to see, because of these new technologies that were spawned by understanding the sequence of the genome, is the ability to, what some people have termed, use personalized medicine to know that your cancer is made up of the following mutations. And I think this is going to be incredibly, it'll change the way we think about cancer. But it also implies that we have another problem. And the other problem is that this is the slide that was up when I first came in. This is a map of the chromosomes for a cancer. Now, I couldn't find a normal one, but there's only supposed to be 2 of these chromosomes in every one of these positions. And what you can see is that there are way too many of them. And for those of you that are close enough, you can see that each of these groupings have different colors. And you can see that there are little bits and pieces of these chromosomes where the colors are wrong. Instead of all being green, there's a little red piece here. Instead of all being pink, there's this turquoise piece in the middle. And the problem is that any given cancer that we find in a patient may have 100 or 1,000 or more mutations in them. And so the problem is, although it'll be great that we can define every cancer based on their mutations, is that there are way too many mutations to allow us to say, one gene caused your cancer. It's going to be, well, one of these 1,032 genes caused your cancer. Now, that seems a little hopeless, and solving problems with 1,000 variables is something that I wouldn't subject any student to. But one of the things that we think is that although there are many hundreds, if not thousands, of mutations in cancers, that there are probably a much smaller set maybe somewhere from 6 to 10, that are really the important ones. And all the rest of them came along for the ride. They're called, and the terms that we're now using for that is that there are driver mutations, the ones that are really important, and then there are passenger mutations. And as in most things, although except on 128, there are many more passengers than drivers. So the problem is once we figure out what all the mutations are, we have to, in an equally robust way, figure out how to identify those that are the drivers versus the ones that are passengers. So going back to the map analogy, for any of you that have bought in a GPS unit now, you know that they don't not only tell you where you are and how to get to where you're going, but they now have this integration of traffic and weather, and they'll try to reroute you around things. They know where the one-way streets are, particularly in Cambridge. And so really, I think what we want to do is we want to move forward in cancer to where we're not only defining the landmarks or the maps and all the mutations in cancer, but we're able to understand the functional consequences of the genes and identify those that are drivers. So how do we do that? And the drivers, in some sense, are the ones that we think are going to be the Achilles heels. And the way we're doing that is by taking a different approach to using the information from the genome. And that is by combining lots of different manipulations, we and others are trying to manipulate genes in experimental models in the lab to try to make cancer cells and try to stop cancer cells. And we're doing this because if you do this in large enough scale, you will eventually figure out all the genes that could potentially cause cancer or be used to stop cancer. Now, I'll go through this in a little bit of detail because it's not necessarily an obvious concept. So one way to think about things is that you could take normal cells and manipulate their genome in the lab and try to make cancer. And actually, a few years ago, my lab was able to do this. And there was a fair amount of excitement about doing it because it allowed people to think about making cancer cells in a very defined way. And in many ways, it's like if you were trying to figure out how to radio work, you could take it apart. And if you can put it back together, you've learned a lot about how the wiring of a radio works. But of course, there were people who were not so happy about that. In fact, there was Jay Leno made the joke that he thought that they were paying scientists to get rid of cancer, not to make more cancer. But that's one way of trying to understand how cancers are built. The other way is something that we call loss of function approaches, and that is you take cancer cells from patients and you try to turn off genes one at a time. And this is a lot like what you might do if you have a fuse box and you're trying to figure out which fuse controls the light that, or your washing machine. You go and you flip the switches on and off and on and off, and you look and see which lights go off. And that's really what we're doing here in the loss of function approach. And what these two approaches are the cornerstone of what molecular biologists have been doing for the last 20 years. But the problem for studying cancer and other diseases is that because before we understood all the genes in the genome, basically we did this process one gene at a time, one person at a time. And so the problem was much more like it would be one student, one gene, and 10 years before you understood what was going on. And if it's true that we're going to find thousands of mutations by studying cancers, that paradigm's not going to work. We don't have enough graduate students to spend enough years to figure out all the functions of those genes. And so what we're doing now is taking these model systems, and because we know all of the genes in the genome, you can actually make what we call libraries or collections of all these genes and introduce them into cells one at a time in a very systematic way. And one can imagine that if you did this in a very systematic way, you can figure out all the combinations of genes when you overexpress them that make a cancer. And so that's one approach that we and others are using to try to define which are the genes that are driver genes in cancer. The other thing that one can do is to use this discovery that was awarded the Nobel Prize a couple of years ago to Craig Mello, who's at UMass, and Andrew Fire, who's at Stanford, is this idea called RNA interference. Now, RNA interference turns out to be something that rewrites the textbooks of biology. The textbooks of biology say that you have genes that get transcribed or made into genes that are DNA get made into RNA, and that RNA then gets turned into a protein that does the work. What RNA interference says is that, in fact, the way you control these genes is by making RNA, which goes back and can turn off genes directly. And while we don't understand all of the implications of RNA interference and why it exists, it is based on the sequence of genes. And since we know the sequence of all the genes, one can use RNA interference in a prospective way to turn off genes at will. And so the Broad Institute, as well as Cold Spring Harbor and a couple of other institutes around the world, have built collections of RNAi reagents that allow scientists to turn off any gene in the genome one at a time. And so systematically, what one can do is turn off genes in the genome and in cancer cell lines and figure out which genes are essential for those cells to survive. And one of the things that we've been doing here is to try to say, well, how can we bring all of this together? So going back to my GPS analogy, we really want to have maps of the genome. We want to have functional data that tells us which genes are important. And we want to put them together in an integrated way that will teach us about cancer. And so we and others are manipulating cancer genes in the two ways I told you, by turning them on one by one and turning them off one by one and looking which ones are important. And we're taking that information and we're connecting it to all the information about what genes are mutated in any given cancer. And the idea here is that if you're both mutated in a cancer and you score in our experiments as being important for a cancer phenotype, you're much more likely to be a gene that's a driver, one that really causes cancer. Now, how does this work? Well, it actually reasonably works well. A former graduate student in my lab did this experiment as a proof of principle, and he identified a bunch of genes that could take cells and turn them into normal cells and turn them into cancer cells. Working with others in my lab, they identified genes that when you turn them off, breast cancer cells died. And the overlap of that, plus the ones that were mutated in breast cancer genomes, identified one gene. And this gene, that has this funny name, IKBKE, is a breast cancer oncogene that's present in 30% of women who have breast cancer. Now, why is that important? It's important because if we have this information, then one can think about making new drugs. And I'm going to give you one example about how this works. And that is a disease called CML. It's a type of leukemia, and a drug that some of you may have heard in the news called Gleevec. So in the 1960s, these two investigators, Noel and Hungerford, working in Philadelphia, discovered that two chromosomes were rearranged, and a piece of chromosome 9 went over to chromosome 22, and vice versa, and made this funny chromosome, which was called the Philadelphia chromosome, because that's where they were. And at the time, they thought it was good to name things by where people were. And this turned out, these two broken pieces made a new gene of a part of a gene that was on chromosome 9 and part of a gene that was on chromosome 22. And after 20 years, people figured out that this abnormal gene, they could make a drug that could turn that abnormal gene off. And this drug was called Gleevec. And over the last sort of 5 or 6 years, it's been shown that if you give patients with this disease that have this specific broken chromosome this drug, what happens is nearly 95% of them are cured of their disease. And there are a couple salient features about this. One is this disease was 100% fatal before this drug was introduced. The second is that the treatment that was commonplace for this disease involved very, very aggressive chemotherapy that made people very sick. And the chemotherapy itself killed 3 to 5% of the patients who were treated and didn't cure the patients with the disease for the most part. Actually, about 90% of the patients with CML. Yeah. And the other thing about this drug is it's a drug that you take 1 pill a day, and it has no side effects. And so compared to what the standard was only less than 10 years ago, this disease has been completely transformed from one that was uniformly lethal, and if you could treat it, was terribly toxic, to one where someone comes in, takes a pill a day, and they have a very high likelihood of not having the disease come back again. Now, some have argued that this is a quirk, an accident, the exception that proves the rule. But I think that most of us believe that understanding mutations like this and figuring out which ones are important will lead to drugs that have a similar outcome as is the case in CML. Now, it's likely to be not just one target, because as I told you, most cancers have thousands of mutations, of which maybe 5 or 10 are important. But even if you had to take 5 pills to take care of your cancer, I think that would be a great improvement on what we're doing now. So if we do these things, I think the whole goal here is to take from where we still are, and as I said, I'm a little bit embarrassed that we're still here in the clinic, of defining cancer by these very primitive ways, is by doing all of these things that I told you about, and here are the fancy names for them, that we're going to redefine cancer by what we'll call its molecular circuitry. And one way of thinking about this is we'd like to be able to draw a map of all the things that cause cancer. This is a map that Bob Weinberg and I drew up a few years ago that's modeled on the London subway. The journal that this was published in is based in London. Otherwise, we would have used the T. But the idea here is that you can come up with these circuit maps. And if you understand the circuit, you should be able to figure out how to turn off the circuit. And the idea that I think we would like is that we would like a drug that stops the red line, a drug that stops the dark blue line, a drug that stops the green line. And I think if we do that and redefine cancer by its circuitry, that we will redefine cancer. We will redefine how we treat cancer. And I think that it's highly likely that we'll change the experience of people who have to go through treatment of cancer and hopefully change the outcomes. And so what I hope, if I were asked to give this lecture again in 5 or 10 years, that I wouldn't start by saying that we use fairly primitive ways to think about cancer, but instead I'd say that we now define cancer based on what molecular alterations occur in any specific cancer, that we have specific biomarkers that tell us who should be treated, who shouldn't be treated, and allow us to diagnose cancers earlier so that they're easier to treat. And although my pathology colleagues would be unhappy, I'd be very happy if we could get rid of this idea of what does the cancer look like under the microscope as a way of figuring out what to do about the cancer. Because I think that although that's a little more sophisticated than figuring out where it started from, it's a concept that is subjective, and we need to move beyond that. And if we do that, I think what we'll accomplish is that we will have earlier diagnoses of cancers, we'll develop improved therapeutics, and we'll be able to decide, and I think this is a very important thing, which patients need to be treated by which drugs, and which patients are most likely to derive benefit from treatment. Because what we do right now is that we have a treatment for a certain type of breast cancer, and we treat all the patients, knowing that only 50% of them actually benefit from it. And so we need to move to the point where we identify that 50% and treat that 50% with the drugs that are going to help them, and the other 50% we treat in a different way. And I think if we do that, then we will have made progress in cancer, and cancer hopefully won't be as much of a public health menace as it is now. And I just want to end with one last point, and that is sometimes I get asked, why does it seem like there's more cancer out there? And the answer is, I don't think the answer is because we haven't been taking care of where we live and that we're exposed to many more toxins. It turns out that we're a victim of our own success in medicine. Because people don't die of infectious diseases, and we have many fewer accidents that killed people than did 50 years ago, and we're much, much better at treating heart disease and stroke, more people are living longer. And the number one risk factor for cancer is age. And so as our population ages, we have more and more cancers that occur. And that's why it seems that there are more cancers in developed countries. And so I think one of the things that we have to think about going forward as we do all of these things is we do want to think again about prevention of cancer and how do we figure out what the connection is between aging and why when people age are they more susceptible to cancer. I suspect it has to do with the fact that as people age, all the things that they've been exposed to over their lifetime and all the copies of the cells that have been made lead to more mutations, and that increases the risk of cancer. But we should think, I think, as we move forward with this, we have to start thinking about how do we prevent those changes from occurring so that we don't have more cancers as people get older. So I'm going to stop here, and I'd be happy to take questions on anything that I presented. I should warn people that there is a reception afterwards, so if we spend lots and lots of time answering questions, the food will wait out there. But I do think it would be good to have some questions, and I'm happy to answer them now. Yes?
I found this driver really very interesting. Not something I'm just curious. It's a very naive question. Could there be an underlying profile of driver genes? And I'm thinking of like the kind of template for body plans in the biology of bilateralism. You have all kinds of diversity. So you have all kinds of cancers, but there must be something simpler about underlying as a principle of this driver gene and identifying that, that would in turn a possibility of future, about diverse range of things. And again, I apologize for the naive question.
No, it's a very good question. So the question is, with the concept of driver genes, do they represent some kind of underlying biological program that if we understood that program, we might understand more about what a driver gene is? And the answer is you're absolutely 100% on the mark. That driver genes the ones that we've discovered so far, tend to be genes that are the normal regulators of growth and proliferation. And so it's when they get out of control and overactive that they push cancer to develop, and that's why we've given them this idea of being a driver gene. Another class of driver genes are genes that are normally the genes that prevent the chromosomes from becoming disorganized. And there are many types of genes that are involved in keeping chromosomes from getting scrambled. And the way you have to think about this is every time you make a copy of each of the cells in your body, you have to copy all the DNA, and all these chromosomes have to make it into the two halves of the new cells. And so there's a lot of mechanisms that are controlling that process. If you break or have a mutation in one of those processes, you then have the capability of scrambling your genome every time you make a copy of it. And that is another type of gene that drives cancer development. And some of you may have heard of the genes BRCA1 and BRCA2. Those breast cancer susceptibility genes are involved in controlling the normal behavior of chromosomes. And that's why people who have those mutations are predisposed to developing cancer at earlier ages, because they don't have as good mechanisms to keep their chromosomes from getting scrambled. So you're exactly on the mark. So you're ready to start in the lab. When can you start?
I also hear somebody mention that DNA methylation also contributes to cancer development. So what's the related importance of DNA methylation as to the DNA mutation?
Okay, so we have a ringer question. So the question is, how does DNA methylation contribute to cancer? So just so I can start here. So all of your blueprints of your cells are made of DNA. The DNA is not passive. There are many enzymatic modifications of that DNA that turn on and off genes and control how the DNA is regulated throughout the life of a person. So DNA methylation is one modification of DNA, and it's thought that what methylation does is it is a signal that tells the cell whether to turn a gene on or off. And so one can imagine that if you muck with the mechanism that controls DNA methylation, all of a sudden you'll turn a lot of genes on that shouldn't be on, and you'll turn a lot of genes off that need to be on, and that could lead to chaos. And what people have determined is that if you look at cancer cells, you find DNA methylation in all the wrong places. And in fact, Rudy Janish's lab, which is next door at the Whitehead, made a mouse that can't turn on DNA methylase. And those mice get cancer at a very high rate. So there is something to modifying DNA, but in many ways I think it's getting back to the question of you're inappropriately turning genes on that shouldn't be on and turning off genes that shouldn't be off, and that's contributing to cancer.
It's scary to hear you say that you can't cure most kinds of cancer, but presumably most kinds of cancer have very small numbers of victims. Am I right in thinking there these downward moving curves on the chart? Am I right in thinking that most cancers, most people who get cancer, doctor can do something effective to cure it?
Yeah, so it is a sobering thought, and one that probably not a good thing to talk about around dinner time or with friends. But so I think what you have to think about in cancer is it goes back to this idea that I presented early on, that cancer is not one disease. It's probably 100 different diseases, and it depends what kind of cancer you're talking about in today's terms. So, for example, we'll start. Do you want the good news or the bad news first?
Start with the good.
Okay, so the good news is if you get prostate cancer, there are about 180,000 people who get diagnosed with prostate cancer each year in the United States. Only 30,000 of those people will die of prostate cancer. So the great majority of people who are diagnosed with prostate cancer will be treated and will survive and live the rest of their lives without worrying about prostate cancer. There's a small fraction that will die. So that's the good news. And another piece of good news is if you have testicular cancer, you have a 95% chance of being cured of testicular cancer. And if you're a kid and you get certain types of leukemia, you have nearly 100% chance of being cured of leukemia. So that's the good news. The bad news is if you have pancreatic cancer, in 2006, there were about 30,000 people diagnosed with pancreatic cancer, and 31,000 people died. So pancreatic cancer is uniformly a fatal disease, and we still can't really impact that yet with all the advances that we've made. So there's some cancers where we've made a lot of progress, and I think another place to look at is breast cancer, where treatment after minimal surgery has definitely saved many, many women and who would have died of breast cancer 25 years ago. But we still have women dying of breast cancer, so we still haven't solved the problem. So it depends a little bit on which cancer you're talking about. And the good news is that I think with all of these tools, many people are trying to get at these problems. Why is it that a pancreatic cancer is so hard to treat, but a testicular cancer seems to be cured so easily? And trying to figure that out, I think it's going to come from understanding what are the changes that underlie those cancers. I think where we're going to learn how this works is by understanding all the changes that underlie each of these different types of cancers. We'll figure out why it's been so easy to treat testicular cancer and so hard to treat pancreatic cancer.
Does everything in the lab has to be done physically with mice and stuff? Do computers and simulations not accelerate some of the process?
Absolutely. So when I started graduate school, you did well, we didn't have laptops and we didn't have computers. That tells you how old I am. So we had to do everything by hand and by paper. Now, no one does anything in the lab without a computer. The data that we can generate in this building would be, we just wouldn't even be able to print it out on a piece of paper each day if we had to. So we are using computers. We are using modeling. We're using statistics as much as possible. But at the end of the day, you still have to show that what you're doing in theory actually occurs in real life. And the same is true of theoretical physicists. Theoretical physicists think about the world and how it's made, and at some point they come up with theories to describe the physical behavior of things in the universe. But they have to go out and test some of those things, and they build these huge super colliders to try to test their theories. And so like any scientist, even though we can come up with theories and models, and that's very important to the work we do, eventually you have to put it to an experimental test. And in this case, when we think about disease, it's incredibly important because you actually have to make a drug. And you have to make something that you can give to someone. And you can't do that in a theoretical way.
What should a cancer patient advocate for or do to help the scientists move forward?
So the question is, what can patients or advocates do to help scientists make progress? I think there are a number of things that advocates can do that have been quite helpful, and some of the things that they're already doing are quite helpful. One is advocates, I think, can be great sources of recruiting patients to join clinical trials. We can't come up with better treatments unless we have patients who agree to be in clinical trials so that we can test and prove that things actually work. Because there's enough variability between each one of us when we take any medication that you need to study enough patients to know that it's working for the large number of people. And I think one example of that is this drug called Herceptin for breast cancer. That trial, which is now an approved drug and is helping a lot of women, would never have been done if the breast cancer advocates hadn't lobbied amongst themselves to ask women to be tested for this and enroll in this clinical trial. So that's very important. The second is that cancer research in this country is largely funded through public support, and the work of the NIH and private foundations fund almost all the work in cancer. The pharmaceutical companies work on cancer, and it's a very small amount because they're not funding basic research. And I think that's probably where we don't want the money for basic research to come, because they have a different agenda. So I think advocates can help cancer research by supporting the initiatives to increase funding for cancer research. Unfortunately, the NIH budget, although it increased dramatically in the late 1990s to 2000, in the last 4 or 5 years has essentially gone down. So overall funding for cancer research has gone down in the last 5 years, despite the problem being just as difficult as it always has been. And despite the fact that many wealthy individuals and foundations have put a lot of money into cancer research. I saw a figure that was sort of staggering to me, and that is that if you combine all public, private, and pharmaceutical support of cancer research, it's about $11 billion. Pepsi-Cola spends all the soft drink companies spend $40 billion in advertising a year. So we spend as a society, we spend 4 times as much advertising soft drinks as we do on cancer research. So I think, although I think the public has been very generous about supporting cancer research through the years, and we're all thankful for that, we are really at a tipping point where we can, I think, in the next decade, make a difference. And this is probably the best time as ever to be pushing that agenda. And finally, I think that what advocates can do is be involved in foundations and get involved in research. There are most cancer centers have strong programs for advocates to come and hear what's going on and to give their opinion about whether they think the scientists are on the right track or the physicians are on the right track, or they're completely off base and they're not hearing the problems that people really think are important. And I think being involved in those groups is very important because it does change the direction of science and what people are studying.
Research on cancer having spillover effects that help with other diseases?
Absolutely. Cancer is a unique, I think the genome-based research is being applied to all diseases. Cancer is a particularly good disease to use this information for because in some ways you can think about the cancer genome as being different from the rest of the body. And we compare everything against what's normal. But as we learn the function of these genes that have gone awry in cancer, we also learn about their normal functions. And some of these genes also are operating in other diseases, such as diabetes or heart disease. And so the more we learn about cancer genes, the more we're going to learn about how normal other genes work in other diseases as well.
On the other side, what about the immune surveillance of the patient? Why is that not working? And is that one reason why cancer increases with age? Is general vaccination patients?
So the question is, what about immune surveillance? Does the immune system systematically try to survey your body for cancer and get rid of it? And is that one reason why when you get older, because your immune system doesn't work as well, people get cancer? I think we don't know the complete answer to that. There have been a couple of experiments of nature that provide some insight into this. And one is that there are diseases where people are born without an immune system, or unfortunately like a disease like HIV, where a disease wipes out the immune system. And it's informative to look at what kinds of cancers those people get. And particularly in HIV patients, they get cancers that are driven by viruses. And so they get certain types of lymphomas, they get certain types of skin cancers, but they don't get lung cancer, and they don't get breast cancer, and they don't get colon cancer, and they don't get prostate cancer. So my bias is that the immune system is quite important, and it might be, and it's certainly playing a role in surveillance against viral-mediated cancers. But it's probably not a major mechanism of controlling cancers in general. However, that's not to say that one can't manipulate the immune system to try to treat patients who have cancer. And so try to use the immune system to trick it into being a surveillance mechanism to get rid of cancers. And I think that there's been some promising results over the last decade about trying to make cancer vaccines that might be effective, a small amount.
What can drugs against cancer target exactly? Do they act directly on genes or on proteins that are created?
Right, so the question is, if you make a drug for treating cancer, does it affect the genes or does it affect the proteins? In general, we attack the proteins. That's if you talk about drugs. There are types of drugs and radiation that attack the DNA and cause problems. But in general, when we're talking about targeted therapies, we're talking about attacking proteins.
How do you manage to not have side effects if you target natural proteins?
Well, so the question is, how do you manage not to get side effects if you target natural proteins? Well, that's the problem with the current chemotherapies, that we're targeting things that are in normal cells and cancer cells, and that's why you get all the side effects of the cancer treatments. The goal, and what Gleevec was one example of, is you're actually targeting only something that exists in the cancer and doesn't exist in the normal situation. And that's why we think that that kind of drug and that kind of therapy has many fewer side effects.
Gleevec, if I'm not mistaken, targets ATP binding sites. But I've been told that actually cancer cells are always mutated. They change the binding site and become drug resistant. So it's also with prostate cancer, secondary hormone therapy. You get relapse they become unresponsive. So how does that pose, how do you find a solution?
Right, so the question is, even with targeted therapies, isn't it possible that the cancer will mutate again and get around that therapy? And I think that that is, in fact, a real problem. And one of the things that I think will be the reason why we have to figure out all the mutations in a cancer and the ones that are important is we need to have not single drug therapies, but multiple drug therapies. Maybe one last question here.
To what extent are cancers caused by single mutations in the sense of single nucleotide polymorphisms, which people around here seem to be better finding, so that you can find something like an RNAi to cause a knockout of that?
Yeah, so the question is, So the question is, how many cancers are caused by a single gene mutation? It turns out that there are relatively few. And those diseases turn out to be easier to treat. And we have pretty good, for many of them, we have good treatments now. The problem is most cancers are like that slide I showed you, that their genomes are completely scrambled, and they don't have one mutation, but they have hundreds of mutations. So unfortunately, that's where we are. That's the reality that we face now. We've gotten the easy ones, the ones where there was a single gene, and we're left confronted with the problem of the cancers that have multiple or many mutations.
Answer everyone's questions. It's really great. You may get more as people